ഈ CME പ്രോഗ്രാം നമ്മൾ കണ്ടിന്യൂ ചെയ്യാണ്. ഡോക്ടർ സിറിൽ ജി ചെറിയാൻ, ഡോക്ടർ ദീപ രണ്ടുപേരെയും ക്ഷണിക്കുകയാണ്.
ഗുഡ് ഈവനിങ് എവരി വൺ. നമ്മുടെ ഇന്നത്തെ 54th സ്റ്റേറ്റ് ആനുവൽ കോൺഫറൻസുമായിട്ട് ബന്ധപ്പെട്ടിട്ടുള്ള നമ്മുടെ ഇന്നത്തെ CME പ്രോഗ്രാം ആരംഭിക്കുകയാണ്. സോ വി ഹാവ് the CME is basically intended to address our COVID-19 scenario, and we have been fighting COVID-19 for last few months. So we have started our vaccination programs, and we have the most eminent faculty in our country, Dr. T. Jacob John, to enlighten us on myths and truths of COVID vaccination. And incidentally, he is the only clinical virologist in India. And now we are facing post-COVID manifestations, and it is highly essential that we should be up to date in that. And we have learned faculty speaking on cardio, respiratory, neurological, and psychiatric issues. Dr. Varghese Spinoza needs no introduction, who is well known as a teacher and clinician. Dr. Arvind is well familiar to all of us, and is a person whom we look forward when there is any management issues. And neurological issues are common, and we have already witnessed. And we have Dr. Dilip R., Associate Professor at GMC Trivandrum, who has been actively involved in treatment of COVID-19 patients. National Institute of Virology, Pune is the apex center of our country, and we have an eminent speaker, Dr. Rima Sahai, who will be taking a session on viral mutation and clinical relevance in the Indian scenario.
Although SMS and other transmission mitigation strategies implemented in most countries during this pandemic have prevented citizens from being infected to some extent, no immunity to SARS-CoV left us susceptible to additional waves of infection. We all accept the fact that the world will not return to its normalcy until safe and effective vaccine becomes available for all and global vaccination program is successfully implemented. As COVID is new to us, the nature of protective immune response is not well understood. So, there are many apprehensions regarding vaccination which are still existing. The law of vaccination coverage points to this, and it's the need of the hour to update all pieces of knowledge from various parts of the country and world. Now we are in phase 1 of COVID vaccination drive, and now we, the healthcare workers, are receiving vaccine. Most of us develop adverse reaction like fever, myalgia, headache, and many symptoms. We are dealing all by our own. But when common public are vaccinated, we need more clarity and need more explanation regarding the most discussed topics like transparency, fast approval, then clinical relevance in view of reinfection and mutant strains. We are lucky to get the most eminent faculties in this field. With pride, I invite Dr. Janardhan Naik, consultant physician, GH Kasaragod, to introduce our faculty. Thank you.
Thank you, Dr. Cyril and Dr. Deepa. Beloved KGMOA President, Dr. Chaskusar, Secretary, Dr. Jayakrishnan, CME Chairman, Dr. Cyril. My dear friends, good evening to all. All of you have come across COVID, post-COVID, COVID syndromes, directly, indirectly. Today we are here to learn more about the COVID vaccines, post-COVID syndromes, and mutations. That's also very important. The world is under the grip of COVID-19 pandemic. This pandemic has brought suffering, fear, death to populations everywhere. Vaccines save millions of lives each year. Vaccines work by training and preparing body's natural immune system to recognize and fight off the viruses and there are currently more than 50 candidates. The apprehensions, fears about COVID-19 vaccines being fueled to great extent by the myths and misconception circulating on social media. To talk on this subject, COVID vaccine and truths, and to clear our doubts, we have eminent speaker, a dedicated teacher with us today, Dr. T. Jacob John, sir. He did his MBBS at Trivandrum Medical College, DCH at CMC Vellore, MRCP at Edinburgh. He is specialized in pediatric infectious disease covering laboratory virology and microbiology in USA. He was Chief Virology Service of CMC Vellore from 1967 to 1995. 28 years he worked as Chief Virology. His discovery of problem of failure of oral polio virus led to Pulse Polio program in India. He led the CMC virology team who identified HIV infection in India in 1986. He continues to be actively involved as chairman or member or advisor to many international and national organizations, including World Health Organization. Dr. T. Jacobson, sir, embodies the spirit of inquiry that leads to scientific discovery and innovation, ultimately leading to healing and alleviation of human sufferings due to disease. With this word, I introduce our professor, dedicated teacher, Dr. T. Jacobson, sir. Over to you, sir. Thank you.
Thank you very much. Am I audible?
Yes, sir.
First of all, my greetings to all the office bearers of the association, Dr. Prasad, Dr. Jayakrishnan, Dr. Cyril Cherian, and especially your chief guest, Rajan Khobragade. My privilege to talk to you very briefly on COVID vaccines. The title was given to me as Myths, Truths, and Common Queries. Can I have the slides? Do you have it? Can somebody project
we
the
can-
slides?
We can share, sir.
This is a tour through the minds of vaccine developers. When the pandemic broke out, many scientists and vaccine companies began developing ideas to make a vaccine. Normally, vaccine development takes many, many years. And here we had to do it in record time. So the urgency was such that the developers of vaccines aimed to have a one-dose vaccine. And a one-dose vaccine has got to be normally a live virus vaccine, but attenuating the coronavirus of COVID was not going to be a realistic option. It may take several years to make an attenuated virus vaccine. So they resorted to the new technologies, although they have not been tried in human vaccines earlier. If you use a live virus as a vector, particularly non-pathogenic adenoviruses, to make sure that it doesn't cause any disease, you go to chimpanzee adenovirus or least pathogenic human adenovirus, types 5 or 26. That is one approach that the Oxford University and the Gamaleya Institute in Russia and a few others tried. The idea is to package in this adenovirus the DNA of the spike protein of the coronavirus. The spike protein's DNA inside the host cell will transcribe mRNA, and the mRNA will translate into spike protein in the cytoplasm. So companies like Pfizer and Moderna asked, why not mRNA itself? But if you inject mRNA, they will not enter the cells. So you have to package mRNA in something that will be taken inside the cytoplasm, and liposomes are the best vehicle. So lipid nanoparticles were constructed with mRNA inside, and once you inject them, the lipid will be absorbed into the cell, and the mRNA gets directly in the cytoplasm. There it is not destroyed very quickly. Their mRNA then translates the protein, spike protein. Why can't you synthesize the antigen and inject it? The problem is to make the antigen and purify in the laboratory is not an easy task. Whereas if you give the DNA or the RNA and the host cell synthesizes the spike protein, it will be absolutely in pure form. Secondly, those RNA, those spike protein molecules will be expressed on the cell surface. And if you express the antigen on the cell surface, then the immune system will respond both with antibodies and T cell immunity. And as you all know, we need both arms of the immunity to be effective against the coronavirus. Single dose did not work. 2 doses were necessary. So some people use the same adenovirus for 2 doses, and some people used a different adenovirus for the second dose. Why not a killed virus? The problem is to have virus grown and inactivated, you need a biosafety level 3 unit. And in India, the only vaccine manufacturer with a biosafety level 3 was Bharat Biotech. They immediately went with culturing the virus, and actually they got the strain from ICMR, and they grew up the virus and inactivated it, and they made killed virus vaccine in a very short time. Killed virus vaccines are usually given in 3 doses. But then, since all of the vaccines were in 2 doses, they tried a 2-dose schedule, and they were quite successful in getting good immunogenicity with 2 doses. They tried 1 week apart, 2 weeks apart, 3 weeks apart, and 4 weeks apart. Finally, 4 weeks apart, 2 doses was the schedule they used in their phase 3 trial. Next slide. Next slide. Okay. The principle of the vaccine and its immune responses are we need both arms, antibody and T cell. Vaccines must elicit both. The host cell receptor is ACE2, as you all know, but the virus ligand is spike protein. So if you exhibit the spike protein, it mostly elicits Th2, that is antibody response. I'm talking about the inactivated virus presenting the spike protein. If you give an aluminum adjuvant, it will enhance only Th2 response. You need Th1 response. So they used a very special novel adjuvant called Algel. Algel is an aluminum salt with an imidazoquinoline attached to the aluminum molecule, and this quinoline has a very special property of acting as an analog of the receptor on the antigen presenting cells like dendritic and macrophages, TLR7 and 8. It's a very interesting adjuvant, and it promotes both Th1 and Th2 pathways. And so they, in their phase 1 and phase 2, they got excellent responses. And both these publications are, one is already in the Lancet, the other one will be coming pretty soon. Next slide. This is the Indian epidemic curve. Can you see the slide? It's a little contrast is low. Our epidemic started somewhere April, May, reached the peak by mid-September, 3rd week of September, and it has been fairly steadily declining. And we have reached in January numbers that are quite close to numbers that we will expect in an endemic state of infection. So we were not able to get the vaccine by September or soon thereafter to cause a precipitous fall. Unfortunately, that did not happen. So we are now coming with the vaccination, but what will the vaccination program do? In order to get the vaccination program rolling, they went to the emergency use authorization like many other countries had done. If we were in a hurry, we could have done this emergency use authorization way back in September itself. Were these approvals ethical? Yes, they had cleared all ethical requirements for emergency use authorization, not for registration of the vaccine or licensing of the vaccine, for which phase 3 trial is a must. The Covishield is undergoing phase 3 trial in India right now, but we have international data based on which emergency use authorization was given. The bridge study is not over, so that if that is over, when that is over, the vaccine may be licensed. The Bharat Biotech's Covaxin, the phase 3 trial is almost over, and its results will be available in March. Only then we can expect the vaccine to be registered or licensed. Where the approvals have been too late? Yes, unfortunately. In 2020, more than 34 countries were already rolling out vaccines. And in 2021, when India's epidemic is fast declining, we are hurrying with the vaccine. So there is a criticism that this vaccine rollout is more political and not a public health choice. I will not go into any more of that criticism or controversy, but let me tell you that it is a humanitarian gesture that the vaccine be made available to the healthcare workers first, and that is what was described as the first phase of vaccine rollout. Next slide. The quality assurance, there was a question about using vaccine vial monitors, which of course ensures that the cold chain was uninterrupted so that the vaccine's potency could be believed to be acceptable. But that's not the problem with our new vaccines, because cold chain maintenance is a physical activity, and we can ensure that the cold chain is maintained well. You don't particularly need VVM on the vials, at least in the beginning. mRNA and adenovirus vaccines have no preservative. Therefore, no open vial policy is allowed. A Covaxin actually has a preservative, 2-phenoxyethanol, and yet the company's current policy is that once you use that vial, then the entire dose should be used up in 8 hours. The packages are 10-dose vials and 20-dose vials. What the company is rolling out now, I believe, is the 20-dose vial. So VVM is only a convenience tool, and its absence does not affect the confidence in vaccine quality. Next question given to me, next slide. Is Covishield or Covaxin better? Well, you cannot easily come to a conclusion that one is better. Both have properties which are mostly positive, but not necessarily. Wide international acceptance of Covishield or its mother vaccine, the AstraZeneca vaccine, is one plus point for Covishield. Its safety, globally, it is passed safety test. Its efficacy, globally, it has passed efficacy, and the efficacy is not great. It's about 70% is probably the best estimate for what we can expect with Covishield in India. Can a third dose be given to increase the efficacy? There is no information, and the vaccine developers were not thinking in terms of a 3-dose schedule. They were only looking for a single dose schedule, and they had to accommodate a second dose. So a third dose is not tested, and so we have no information. And because it's a live virus vector, the immunity to the vector may interfere with the third dose. Therefore, the third dose is not going to be a good idea with Covishield. Covaxin is an Indian product. safety probably better than Covishield because the adverse events are very few and no neurological events like Covishield had to face has occurred with Covaxin. The efficacy is yet to be evaluated, but the immunogenicity is quite good. If the immunogenicity is moderate, let us say equal to Covishield, then a booster is very possible as a third dose. In fact, the company is planning to have a 3-dose schedule if the regulatory agency agrees with that idea. If revaccination is needed in future, like for influenza, again, Covaxin will win over Covishield. And therefore, these are the plus points of Covaxin. Next slide. Another question was about giving the vaccine in pregnancy and in childhood. There have been no studies and no data. The Bharat Biotech has started Covaxin trials in children, and so we will know soon. There is no study in pregnant women of any vaccine. The kill virus vaccine should be safe in pregnancy. In USA, using the mRNA vaccine, scientific opinion is now gathering to promote vaccination in pregnancy because they are seeing some severe disease in pregnant women. So pregnancy is probably a risk factor for severe disease. Therefore, that process of thinking is going on in the United States. Next slide. Is the vaccine safe in those who have recovered from COVID or asymptomatic coronavirus infection? The question arises because many healthcare workers have been infected. There is no particular study and no particular data, but in many trials, those who are accidentally detected as already immune, there have been no safety issues. Theoretically, there should be no problem. Vaccine will be boosting immunity as in influenza vaccination. So we don't expect any problem of vaccinating people blindly. Next slide. How long will immunity last? Only time will tell. The infection-induced immunity seems to wane over months. It is expected to last at least a year as a protective efficacy. I'm talking about antibody levels, they decline, whereas cell-mediated immunity is likely to last long. All respiratory infections like flu, you need an annual vaccination. Whereas other respiratory infections like measles, once you're immune, your immunity is lifelong. COVID may be in between, in which case you may not need annual vaccination, but you may need periodic vaccination, in which case you need a vaccine that can be repeated. And as far as I can predict, mRNA vaccine and kill virus vaccine should be okay. Vectored vaccines with adenovirus may not be okay because the immunity to the vector virus will interfere with immune stimulus. Next slide. Allergies and vaccination. Very recently, that has come up in the media. Anaphylaxis with Pfizer vaccine may be due to the fact that it contains polyethylene glycol. Moderna does not contain polyethylene glycol, and no other vaccine contains polyethylene glycol. Therefore, anaphylaxis may not be a problem, but then anaphylaxis can occur in anybody with any vaccine. There is a question whether the liposome that are being used could induce anaphylaxis, and that is a question that we need to ask and answer. However, anyone who gets any injected vaccine should remain in the premises for at least half an hour to make sure that there is no anaphylactic reaction. Anaphylaxis is more common in the West than in India. Still, we have to be careful, and anaphylaxis treatment like epinephrine should be available within reach in all immunization stations. The That kind of reaction is unlikely with kill virus vaccine. Immediate allergy is anaphylaxis. Delayed allergy is something that we are not very much worried about. The Pfizer vaccine has been found to have some allergic reaction, delayed allergy, but no other vaccine has been shown to have delayed allergy. So allergy is not a contraindication, but we have to make sure that you follow up after taking the vaccine for any allergic reactions. No life-threatening allergies anticipated except for the polyethylene glycol containing vaccine. Next slide. Viral mutations in vaccine, another talk is coming, therefore I will not spend much time. Most mutations have not affected the fitness of antibody, especially if the antibody is polyclonal. If it is monoclonal, yes, some mutations have affected the polyclonal fit with the antibody. The South African mutants seem to be less protected by convalescent serum antibody, and therefore we need to be careful. The future is uncertain. Viral mutations are not altering the ligand that fits with ACE2, the receptor binding domain. Therefore, it's unlikely that these mutations will affect immunity in a major way. All the mutants are currently called variants, not new strains. Will we have new strains or vaccination escape mutants? We do not know. The future is uncertain. But so far, the mutations have not been a problem in protection by vaccines. Next, why were healthcare staff targeted for vaccine rollout first? The government has not stated why, raising these controversies that I mentioned earlier. Is it a humanitarian gesture? Is it to confirm safety in the sense that healthcare workers being vaccinated, they will be the first to pick up any safety issues when the vaccine is rolled out in a major way? Vaccine is being rolled out not with registration, but only with emergency use authorization. Or the accusation from some quarters is that it's a political rollout, because the claim was India will have the world's largest rollout. So these are the questions that I got, and I thank you very much for your patient listening. If there are any other questions, I'll be happy to answer if time permits.
Thank you. Thank you very much, sir. We have a few more questions to answer, sir. Shall I ask?
Please do.
If someone gets COVID after first dose of vaccination, when should the second dose and any need of antibody test? If so,
Yeah,
at
if the
which time?
if the COVID is confirmed, that itself will boost the immune response from the first dose. First dose is partially protective, so therefore the disease is most likely to be non-severe. That person does not need a vaccine in a hurry. Perhaps you could wait a year before you give another dose. No hurry for a second dose.
Sir, there was a question in the chat box. If somebody gets COVID after the first dose,
Yeah.
is it significant or?
No, that's likely to be less severe because of partial protection.
Sir, one more question. The route of vaccination, is there any chance of respiratory mucosal route in the immediate future?
Yes, not immediate future, but in the near future. There are companies that are looking at nasal vaccines. The advantage of nasal vaccine is that you could induce very high levels of local mucosal nasal immunity, and that could help in interrupting transmission. Why do you think of interrupting transmission now? Because this is not a good virus to have around in human communities. And if it can be eradicated, nothing like it. If you don't eradicate this virus, there are two biological problems. One, we already have heard. Can mutations make the virus misfit for vaccines, and therefore you have to scramble for new vaccines? Before that happens, we should be able to eradicate. The other problem is a bit sinister. Now we know that cat family, cats, tigers, lions, puma, are susceptible, but they don't seem to reinfect or back infect humans. That's out of the problem. Dogs and dog family, similarly, they get infected, but they don't transmit infection back. But the Mustelidae, that is mink and related family, related animals, both farmed mink and wild mink have been infected, and mink has transmitted infection back to humans. So before such animals become another reservoir of this virus, we must eradicate the coronavirus. And newest in that list is farmed deer. Now, deer is Cervidae, and they are ruminant family, and they are very susceptible for infection, and they are quite likely to shed large amounts of virus in their respiratory tract. So before such animals become a new reservoir, we should aim for eradication, and a nasal vaccine would be very handy, particularly given as nasal drops, just like oral drops for an eradication drive. So yes, vaccine developers are thinking that way ahead.
Sir, the questions won't end, it seems. One last question: is there a need to check the antibody titer before and after vaccination? And if there is a, if so, what is the ideal time? And another thing, if the under 18 cohort is excluded from the vaccination, will there be a viral preference for that age group later on?
The virus will not know who is immune, who is not immune when the virus attempts to infect. So that's not a problem. Whereas below 18, although their severity of disease is low, they are good vectors of transmission, particularly children above 10. Children below 10 are not good transmitters, but occasionally they get very severe multi-system inflammatory disease. So the Bharat Biotech has started a vaccine trial in children from 2 years upwards. So we will know once their studies are over whether children can be vaccinated. Until then, children are not a target for vaccination in any country. And by protecting adults, they think that the problem can be solved. But in case we move towards eradication, we will have to vaccinate a broader age group. Therefore, children also will have to be targeted. And the inactivated virus vaccines and the messenger RNA vaccines are theoretically highly safe, whereas the adenovirus vector vaccine, we have to prove that it is safe. So that is all we know today.
Thank you. Thank you, sir. There are a few more questions, but due to time constraint, we are moving on to next topic.
Okay.
And
Yeah.
we hope that you will be kind enough to answer the questions in the chat box itself, sir.